Greetings and welcome to the United States Transhumanist Party Virtual Enlightenment Salon. My name is Gennady Stolyarov II, and I am the chairman of the U.S. Transhumanist Party. Here we hold conversations with some of the world's leading thinkers in longevity, science, technology, philosophy, and politics. Like the philosophers of the Age of Enlightenment, we aim to connect every field of human endeavor and arrive at new insights to achieve longer lives, greater rationality, and the progress of our civilization.
Vamos a lanzar esta iniciativa para el mundo, la declaración de Madrid por la longevidad, en 3 lugares. Aquí, en la presidencia de la Comunidad de Madrid. Luego iremos al Congreso de Diputados, a la Plaza de las Cortes, y terminaremos en el Ayuntamiento de Madrid, en el Palacio de Cibeles. Nuestro objetivo es ¡viva la vida! ¡Fuera la muerte! Tenemos la declaración que vamos a poner en internet, la declaración de Madrid por la longevidad. ¡Viva la vida! ¡Muere la muerte! ¡Muere la muerte! ¡Muérete! 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 Pon la cabeza arriba, la cabeza arriba, la cabeza arriba.
I think he needs to help us. He's not helping us now. Save him, save him.
¡Viva la vida! ¡Fuera la muerte! Oye, se te perdió el palo. Yo lo zafo, ¿verdad? Ok. Ahora queremos leído la declaración de Madrid por la longevidad. Vamos a la segunda parada. La segunda parada es el Congreso de Diputados. Agárralo. Ok, vamos a ir ahora al Congreso de Diputados. Todos los políticos de España tienen que saber que estamos cerca de curar el envejecimiento. Cerca de curar el envejecimiento. ¡Stop envejecimiento! ¡Stop envejecimiento! ¡Stop envejecimiento! ¡Stop envejecimiento! ¡Stop envejecimiento! Ahora vamos, sigamos. Déjame. ¡Viva la vida! ¡Viva! ¡Viva España!
He's my hero. I'm voting for president of the European Union. Entre la última generación mortal y la primera generación inmortal. ¿Dónde quieres estar tú? ¿Entre los últimos que se mueren o los primeros inmortales? ¡Inmortal! 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 ¡Inmortal!
nombre de uno de los partidos políticos que apoya el transhumanismo, Alianza Futurista. Fuimos el primer partido transhumanista que se hizo en España y unos primeros del mundo. Nosotros no queremos nada de la lucha entre partidos de la derecha y de la izquierda. No queremos jugar dentro de ese juego. Nosotros lo que buscamos es el bien de la humanidad, acabar no con la muerte, porque en realidad es imposible, pero sí prolongar la vida todo lo que podamos. Y para eso necesitamos que se actúe. Y la manera de actuar es evidentemente metiendo dinero en la información y para el público y en la investigación. Necesitamos investigación, dinero para investigar contra la vejez. Así que apoyo de todos para este objetivo que tanto buscamos. Y por supuesto, uniros a nosotros, porque nosotros lo que buscamos es, ante todo, el bien común, el bien de la humanidad, de toda la humanidad. Gracias.
Ok, 1 de octubre, Día Internacional de la Longevidad. Muerte, vente para acá. Muerte, vente, que te vamos a matar. Vente. Pórtate bien. Ok, ¡viva la vida! ¡Viva! ¡Muere la muerte! ¡Muere! ¡Viva la vida! ¡Viva! ¡Muere la muerte! ¡Muere! ¡Muerte! ¡Vamos a matarte! ¡Muerte! ¡No! ¡Vamos a matarte! Alguien que la mate, mátale. Eso. ¡Muere la muerte! 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 ¡Viva! ¡Viva Madrid! ¡Viva! Stop aging! 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 Stop envejecimiento! 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 Stop ¡viva! ¡Muera! ¡Viva! ¡Viva! Stop aging! 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 Stop envejecimiento! 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 Estamos aquí, 1 de octubre, que es el Día Internacional de la Longevidad, para celebrar la vida de todos, la vida saludable, la vida más joven, la vida más larga, la vida más y más para todos. Y por eso hoy, 1 de octubre, aquí, frente al Ayuntamiento de Madrid, en el Palacio de Cibeles, pedimos todos juntos vivir más, vivir mejor, vivir más tiempo, vivir más sano. ¡Viva la vida! ¡Viva! ¡Fuera la muerte! ¡Viva! ¡Viva Madrid! ¡Viva! ¡Viva España! ¡Viva! España es el país más longevo de la Unión Europea y Madrid es la ciudad más longeva de toda la Unión Europea. 86.1 años de expectativa de vida en Madrid. ¡Viva Madrid! ¡Viva! ¡Viva España! ¡Viva! ¡Viva la vida! ¡Viva! ¡Fuera la muerte! ¡Fuera! 1 de octubre, Día Internacional de la Longevidad. Estamos aquí para matar a la muerte. ¡Muerte! ¡Ven para acá! ¡Muerte! ¡Ven para acá! ¡Ven, ven, ven! ¡Ven para acá! ¡Muerte! ¡Muerte! ¡Vente! ¡La muerte! 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 ¡La muerte!
La muerte tiene miedo, la muerte tiene miedo.
Nosotros queremos vivir y te vamos a matar muerte. ¡Viva la vida! ¡Viva! ¡Fuera la muerte! ¡Viva Madrid! ¡Viva! ¡Viva España! ¡Viva! ¡Muerte! ¡Ven para acá, muerte! ¡Te vamos a matar, muerte! ¡Muerte! ¡Al paredón! ¡Matar a la muerte! ¿Quién mata a la muerte?
¡Te matamos ya! ¡Ven por aquí!
Que alguien le pinche a la muerte. ¿Quién tiene para pinchar?
¡Ven por aquí!
¡Ven, ven! ¡Eso, eso, eso! ¡Muerte a la muerte! ¡Fuera la muerte! ¡Fuera la muerte! ¡Fuera la muerte! 1 de octubre, Día Internacional de la Longevidad. Estamos aquí por la vida, más vida. ¡Viva la vida! ¡Fuera la muerte! ¡Viva Madrid! ¡Viva España! ¡Hay que matar a la muerte! ¡Fuera la muerte!
¡Stop envejecimiento!
¡Stop envejecimiento! ¡Stop envejecimiento! ¡Stop envejecimiento! ¡Stop envejecimiento! ¡Stop
¡Fuera la
Aging is a natural process.
But it's a process which helps our health and well-being.
We are lucky to have longevity researchers developing rejuvenation therapies.
Welcome to TransVision 2025.
So longevity is, of course, not just a theory, it's a biology. And in our next session, we're going to explore the cellular model of age, really decoding how exercise, immune resistance, and epigenetics really shape our health span or lifespan. And we're going to hear from leaders who are really mapping aging from the molecule to the system level and what it means for interventions today. So we're going to have speakers who are going to talk 20 minutes each, and then at the end, we're going to have a Q&A. So make sure either if you send in your questions, your Slido, or that you have some questions prepped afterwards. And first up, we have Vijay Vaswani, co-founder and CEO of Omniscope, together with Holger Heyn, co-founder and CSO of Omniscope. And they are both innovators in single-cell and multi-omics, building maps of how aging manifests in our biology, really on a mission to crack the so-called immune code. So Omniscope, just shortly, is decoding the immune system and using AI to scalably develop diagnostics and accelerate therapeutics, building the world's largest immune data bank. So please give it up for Vijay and Holger.
You are made of cells. All of you are made of cells. Not all cells are created equal. Your immune cells are what are defending you and protecting you from disease right now. They are both the detectors and the very living medicine that allows you to live a long and healthy life.
In the end, it's our immune system that keeps us healthy. Especially here in Madrid, people live healthier, longer lives, and we want to figure out why that is. So in age, specifically, the immune system is losing its function. So we lose protection from viruses, but we also lose protection from deadly diseases such as cancer. Immune cells also lose their self-control. And here they attack our healthy tissue and really make us suffer diseases such as autoimmune or neuroinflammatory diseases. So at Omniscope, we are reading the immune system. We implement a technology that reads the immune system one cell at a time. And that now, over the years, really allowed us to generate a data bank, really to understand the holistic immune system and how it acts in the healthy context, but also to use that as a reference to quantify what's going wrong in disease and what's going wrong while we age or suffer age-related diseases.
Indeed, when you think about the fundamental mechanisms that rule our biology, you could think of the brain as a central command system and the immune system as your distributed cellular soldiers. Now, decoding the immune system is not a small feat. It is an incredibly complex system. And there is not much utility in only being able to understand something if you cannot act. So not only does Omniscope read the immune system at millions and millions of cells, it writes the immune system. We write your immune system so that you can use your own immune system as a living medicine. And this is not science fiction. We've actually done this.
So we have the entire system vertically integrated, where we're reading the immune system and specifically immune system dynamics when, for example, we age, or when patients respond to treatment, for example, immunomodulating drug, immunotherapy, cancer vaccines. And here we also implement a system where we biobank healthy cells and to use those for future autologous therapy. But what you see here on the screen is actually the next phase. We are writing the immune system. In this case, we are writing the immune system of a half-a-year-old child that was suffering from a pediatric tumor. So we are working with her over the years now, she's on complete remission because she responded to immunotherapy. It's a drug that is activating the immune system. And as a company, we were following that process and we were identifying those cells that were killing the tumor cells and that were protecting the patient from the tumor coming back. So it is and it was the immune system that prevented her cancer to come back. She's still in complete remission now after 3 years. But as a company, we kind of took the language of the immune system. We took her own T cells and we engineered the T cells to recognize the tumor. And this is something that in the end is a living drug and will protect her tumor for coming back in the future.
So it's really the ability to use your own biology scalably, and scale is very important. You see, engineering your own immune system the way we've done here, using mRNA technologies, was not only fast, it was incredibly cost effective. And it was cost effective for a public health system. And at Omniscope, we believe that healthcare is for everyone. This key point, moving from immune engineering to the fact that in order to engineer the immune system and to bring healthcare for everyone everywhere, you need to represent everybody cell by cell. That's billions and trillions. You yourself have over 2 kilos of T cell dry weight within your body, up to 10 to the 16 unique combinations. And Omniscope really understands that in order to use data actionably and sustainably, we need to represent all of you, which is why we've spent 5 years building the world's largest immune data bank.
So having sequenced millions of cells from thousands of individuals in the end really allowed us to work with drug companies to develop better drug, to increase the drug development cycle, to make it faster. We were working in areas of cancer, mainly immuno-oncology, infectious disease, but also autoimmune diseases, really where we could make an impact by reading the immune system, sometimes in real time working with clinical researchers in clinical trials. But what about aging? What about being healthy? So having sequenced thousands of samples from newborns to centenarians really allowed us to understand the process of aging and to use our system as a biomarker to quantify the process while we age, or to quantify the process while we respond, for example, to gerotherapeutic agents.
And as my esteemed compatriot, Dr. Mahmoud Khan, alluded, this is not about a single nation or a single individual or an act of heroism. This is about really all of humanity. So we not only looked at different age ranges from newborns to centenarians, we also looked at different HLA types and ethnicities. We also ensured that our models are equally representative from a sex perspective, because whilst our biologies as humans are similar, our sexual morphologies and differences do mean that we react to age differently. It's really this database that really allows us to have a foundational model of the immune system. And really moving forward, it is only by building a model that you can truly act. You can only act on what you know, and you can only de-risk and intervene if you can predict and model what may happen. Thus, by building a complete model of human aging at a cell-by-cell basis, not only do we bring that to you so that you can live longer, healthier lives, we bring it to our esteemed colleagues developing therapeutics as well, so that they can use these models to test and accelerate the ability to enhance human lifespan and quality of life.
So during the next days in this conference, you will hear a lot about the hallmarks of aging. But in the end, it is the immune system that is either the root cause or an actual consequence of these hallmarks. So if, as a company, we put together a model of age that is incorporating our own proprietary sequencing methods, our proprietary machine learning models, for example, related to immune age or the inflammation process. But we also pair that up with already existing, with accepted markers biomarkers of aging, for example, senescence, telomere length, or the DNA methylation clock. And I'm obviously very proud to have Steve Horvath in this session, who was the pioneer looking at methylation and the prediction of biological age.
And it is also an honor for us to use some of the technology which Dr. Horvath has designed. In science, we genuinely mean it when we say we stand on the shoulders of giants. And I do want to take a second to thank you for being such a major contributor to our field. Thank you. Thus, we build a model of age with respect to what has come before us scientifically, but also moving the field forward with all of the tremendous advances. How do we use this model of age? You see, now that you have a model, you can genuinely, from a public health and epidemiology perspective, look at age as a disease. Now, you know, we often look at aging and say, well, all the bad stuff happens to us when we age. From a incidence perspective, as we age, cancer, cardiovascular disease, susceptibility to infection, all become more and more prevalent. And one of the fundamental reasons for that is an aging immune system. But I also want to take a step back. Being mortal isn't all that horrible. Other good things happen to you when you age. You become a little bit smarter. You have families. You can enjoy your wealth. You can give your wealth to better causes. You can use that knowledge to make a better world. So for us, from a humanistic point of view, we want to extend your health span so you can do more good and not just get a lot sicker. So aging as a disease is something which we show in our next slide that is not always entirely bad news. You see, I am interested in you, but I'm going to take a moment to talk about me. My name is Vijay, and I'm the CEO and co-founder of Omniscope. This is my cellular avatar. This is my actual, real cellular avatar being updated right now with my wearables, and is a digital model and representation of my own biology with millions and millions and millions of cells. It is as personalized gets.
So my name is Holger Heyn. I'm Chief Scientific Officer and co-founder of Omniscope, together with Vijay and Lynette, who's here in the audience. And this is my cellular avatar. And my question is, being here in a blue zone, how are these cellular avatars actually changing during the next days of the conference? And as you can see by Vijay and my own avatars, they look very different. And we explain in the next slides what that different actually means and how we challenge each other to get younger or older related to inflammation or the aging process in general.
So let's do the riskiest thing that you can possibly do at a conference. Let's go for a live demo of OS Lifetime, which actually allows us to look at my own immune system. So, going to the live demo of OS Lifetime, which my colleague Nina is just about to switch over to. OS Lifetime is really designed for you and for science-based longevity clinics. We believe in a world where AI and software have a physician at their helm. Anina, if you could move to the demo. If not, I'll admit defeat at my only chance at doing a Steve Jobs, but I guess I'm not going to do that, am I? One of my major issues that I faced, which you can see in my previous slide, is whilst my date of birth is 42, my immune age is actually 60. Whilst I have practiced sports at a semi-professional level for many, many years, one of the things that I unfortunately suffer from is psoriasis, quite acutely. I inherited this as a gift from my father, although he gave me a lot more good things than just psoriasis. And the issue that I've had is that my inflammation at one stage, when I compared it to what we had in our database, was actually as high as a cancer patient. Now, for a CEO trying to spend their life helping other people, that was not necessarily the best message I got. So what you can really see here is that now my inflammaging is back to normal. Indeed, if I actually go to my actual inflammaging, you can actually see how bad this got. The peaks and troughs that you see going up and down are my psoriasis flares. And the reason that they go down is because of administration of corticoids, which are a synthetic form of cortisol, a stress hormone, which has both pro-inflammatory and anti-inflammatory effects. Unfortunately, like many patients after over a decade of use, I have resistance to this therapy, and it doesn't really work. And psoriasis doesn't just attack your dermis, your skin, it can get to your bones. That's not the end which I wish for myself. And for this reason, I actually then also realized as we scroll down that not only was my inflammation off the charts, my cellular damage was off the charts. And not only am I shorter than Holger, my telomeres are shorter than Holger as well. And my telomeres are what allow my cells to constantly actually replicate. And as they get shorter, the ability for me to regenerate my cellular populations is impaired. I can also see that aside from my immune age, my methylation clock also shows signs of accumulated, accelerated aging. But let's move for a second to the presentation where I can show you very briefly what I did to reduce my inflammation. You see, I decided to use non-pharmaceutical interventions. Why? Because I wanted to contribute as an N of 1 to society and show you that you don't always need access to expensive medical care to actually change your biology. I did stuff which is quite free. I slept. I worked out. Thanks to Football Club Barcelona, I changed the way I work out, and we'll talk more about that because as you age, you need to exercise, but you need to change the way you do it. And I took supplements which are generic and cheap. Fish oil, melatonin, high-dose vitamin C. I'm publishing the entire thing, and it's completely free. Don't do it without medical advice, but here's what happened to me. You see, in the next slide, you can actually see quite clearly that in just a few weeks, I went from cancer patient inflammaging to athlete fit inflammaging. And this was a major, major benefit to me. I want to make sure that using OS Lifetime, you have the same benefit. If you can see it, track it, you can do something about it.
And as initially introduced, so our technology is reading single cells. It's reading single immune cells. So we can look, switching to the next slide, really to look at the root cause. What is behind? What's the mechanism behind the improved inflammaging score? So here we're looking at very specific parts of the immune system, CD4 and CD8 T cells, but those can also produce inflammatory markers. They're not only helping us, defending us against pathogens, but they can be pro-inflammatory and produce inflammatory markers that we measure in the blood and that cause systematic damage of organs. So we've seen then with correlation of the reduction of the inflammatory scores and inflammaging, those immune cells also going down. And this is something obviously we are tracking now over time. Going to the next slide, let me introduce you my own cellular avatar. So here you can see in one single view, inflammaging immune age protection score. And what you can see from my immune age that although I'm 46-year-old, my immune age is actually 37. So this is very good news, and this is something I will follow up. I will try to keep improving. Bad news, though, is that my inflammaging is very high. But the good news again is it's something actionable, right? Together with my protection score, displayed by the rings around the sphere, points to the fact I have a solid protection. But again, protection is cellular defense against pathogens, and that's something actionable, for example, using vaccination. Next slide, please. So here today I will walk you through, starting with the view today. It's how my immune age and inflammation looks like, to take you back in time and how the evolution of the immune age has been over the last almost 2 years. So you see kind of a constant gradient, consistent profile over the last 1.5 years, but then at the end of 2025, I was facing a disease called Lyme's disease, which is introduced through a tick bite, then bacteria causing inflammation. It can have a systemic effect. So you see actually the drop in my immune age. You see through the integration of my variables, the drop in VO2 max. And all this together was a warning signal. Then quickly the disease was resolved using high-dose doxycycline. And you see actually both immune age, VO2 max going back to baseline. Now, thinking about it, it's a bit counterintuitive, right? So I'm facing a disease, still my immune age is decreasing. So I'm actually decelerating, I'm getting younger. But if you think about it from the other angle, from the kind of defense immune biology angle, it is really through a disease that is quickly resolved, we are training the immune system. It's a little bit like a vaccine, right? So we're training the immune system, we make it younger, we make it better, we make it defending us better in the future. And this is what has happening. And again, looking at the immune age model as a general, we can then look at specific populations, again, identifying CD4 T cells that are very much correlating while my immune system is getting younger and I'm responding back to baseline. These CD4 T helper cells, so those cells that are building a stable memory, in this case against the Lyme's disease pathogen, are going back to baseline. But then having the full model of age, we can look at other biomarkers of age. So next slide, please. So here, a biomarker senescence, so zombie cells that cause chronic inflammation, are actually behaving the opposite way. So during Lyme's disease, senescence is peaking, and then it's decreasing while I recover, and my immune age is going back to baseline. Next slide, please. Slide before, please. One important thing to mention is, and it's where many aging clock fail, is reproducibility. So if I send a sample, I measure it 3 times, it should give 3 times the same result. And this is what you see on the slide on the right-hand side, where we measured multiple samples repeated times, and it gave, you can kind of see that by the low standard deviations, it gave repeatedly the same result. But then we also looked at biological replication. So samples that you see in the circles, last slide please, that you see in the circles where I have been giving blood repeatedly over weeks. So these are samples taken only weeks apart. And here, the immune age prediction is really following the evolution of my immune age over time while I was recovering from Lyme's disease. What you see here is really then now not only Vijay and myself, but we now are testing many individuals for years now, and you see the consistent evolution of immune age over time. But in specific cases, for example, myself suffering from diseases, you see an active training, or for example, Vijay, you see active peaks where his psoriasis flares are really acting against the immune aging clock.
So really the message here is that we've been very, very rigorous at understanding aging as a process across thousands of individuals from different age gradients, different nationalities, healthy, affected by disease, in a reproducible manner to really measure the way our age changes, both to infection, to disease, but also very important to therapeutics, as well as other interventions. But ultimately, we also need to understand, how do we compare? A model allows you to compare yourself to other people. It allows you to compare yourself to your aspiration. In the next slide, we really go into what is it that we can learn from long-lived centenarians. Omniscope has invested a significant amount of resources thanks to our investors who are here in the audience today, to really understand that. And one of the things which José has mentioned, which I'm entirely aligned with, is that Spain itself is also quite a special country. It's a country where many long-lived individuals do live. What can we learn from these individuals? What can we learn from centenarians, Holger?
It's centenarians, or in this case, even super centenarians. So what you see on the screen is a study that was published about a week ago. It's a multi-omic profile. That basically means there are different layers of information, looking at the microbiome, looking at immune age clocks, looking at DNA methylation clocks. So everything kind of summed up with a holistic profile, how a super centenarian was managing to live that long. And this is obviously our North Star. And this is something what we implement in our lifetime. So you and myself, I can do today already. I can track how my specific biomarkers of age are on track to be a centenarian or potentially super centenarian in the future. And that's what we are all after, right?
So centenarians are the North Star, but we also have to have an idea of where do we want to get to, right? So for a lot of us, as we age, we want to retain our functionality. We want to be able to play. We want to be able to move. We want to be independent. These are hallmarks of vigor and of leading a complete life. Do you know which population has all of this vigor? If you think of it, if you think of the individuals you know who are most functional, in our next slide, it's athletes. Athletes represent, arguably, a paradigm in what it means to be fully functional, fully vigorous. They have less sickness, they have more activity, they keep more muscle mass, they suffer less sarcopenia, they keep cognitive vigor, they are engaged with their communities and their teams. They are brimming, simply brimming with life. And we live in a world where centenarians are running 100 meters and competing in incredible athletic events. If there's one common sense thing that we ought to do, it is to learn the secrets of athletes. And it is with great privilege that we have Dr. Gil Rodas from Football Club Barcelona, who is going to share with us the secrets of athletes.
Thank you very much. First of all, I would like to talk with a microphone. Is this possible? Oh, he's here. I'm sorry. I didn't see. Hola? 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 Fantastic. Well, first of all, I would like to say thank you very much to the organizers for this great event, and especially, of course, to Omniscope. Why I am here? From Football Club Barcelona, in Madrid. Well, I am a privilege because I am working on probably one of the best football clubs in the world, but at the same time, I am a privilege because I am working also in one of the best hospitals in Europe, that is Hospital Clinic and Hospital Sant Joan de Déu of Barcelona. For this is nice in order to share this double vision from sports to the health, to the sports, okay? I am a sports physician, and now I believe that you are a little bit tired, because after 1 hour and a half or 2 hours sit down, please, I think that it will be fantastic if you want to stand up and move a little bit. Yes, fantastic. How many people are suffering low back pain now? Because no? Thank you very much. Okay, sit down, please. Well, next slide. Why we are here? It's true that during many years I have been researching with these guys, with these athletes. No. And these athletes are really different, sincerely. I had the privilege to be their sports physician during many years, from Messi, Iniesta, and Ronaldinho. And believe me, they are different. They have some special things that we need to know. Not only in terms of the genes, if not in terms of how they adapt depending on the epigenetic factors. And this is a great secret that I can have the possibility to know it. Not just only in terms that to help Football Club Barcelona to be greater than Real Madrid or Athletic de Madrid, but also for the future for the general population. And this is a question for you. And I hope that you answer me at the end of this short speech. What do you believe if I say that these athletes probably will have longer lifespan and better health span than all of you? Do you believe that this is true or no? Please, wait a minute. I have the answer, the scientific answer, but please, that is true that everyone in the world think that this is not good. Well, next slide, please. Of course, the cancer in the world of sports is injuries. Yesterday, Real Madrid and Athletic de Madrid won. That is fantastic, but as you know, there are many players that have suffered different kinds of injuries. And our cancer are injuries. Our players are constantly in an inflammation process. All the players that played yesterday, today have inflammation. And you can imagine that this, during 20 years, 25 years of your life, every day you are suffering an inflammation. For this reason, when we, as doctors, we need to know more about the health of our players and to find a good biomarkers in order to help the health of my players and at the same time increase the performance of my players, I found Omniscope. Because it's the first time that I see something that can help me in order to know the immunologic status of my players. Next slide, please. This is a dream or no is a dream. Today, we create, thanks to these partnerships, a good avatars of every of my players. Why? Because I need to anticipate when this player will suffer another injury, or when this player will suffer an increase or a decrease of his performance. For this reason, today, we have the possibility to create this avatar using the digital twins technology. Repeat, this is not the future. I'm sorry, this is the present. Next slide, please. In terms of, that is difficult sometimes, we need big data of every of our players. In terms of from omics, we are talking about the genes, transcriptomic, proteomic, metabolomic, epigenetics, also immunologic profile. But we can incorporate and integrate radiomics, GPS, because we have the possibility to know exactly the load of our players during all their career. Load is very important because they need to adapt every day to the training. You can imagine it today to talk with the players of the Real Madrid and say, how are you? I am tired because yesterday I played. And how is your muscle? Is okay? You're hurt. How is your immunity profile now? We need to know this. It's true that sometimes the players don't like that you know everything about them because maybe yesterday, after the match, they went to the party. But repeat, we need to know this because probably this weekend, when they play again, some of these players will suffer injury. And then is when the coach say, hey, what's happened here? What's happened? If you allow that these players play every 3 days, probably you will have injury. Well, thanks to this kind of universal biomedical, we have the possibility in terms that to anticipate. Next slide. Thanks to Omniscope Sports, we are creating this. This is possible. And this is sometimes is more difficult to convince to the coach that this tool can let him to have more information. Finally, the decision is from the coach. The coach is the man in the world of the sport world. In the hospital is a doctor, but in the sport is the coach. Coach is the boss. But coach needs to believe in this kind of tools. But for this reason, these tools need to be very rigorous. Next slide, please. We create these profiles. We have these tools. Of course, the doctors are very, very happy with these tools. Repeat, every day we are increasing the information about every of our players. Next slide, please. This is very interesting, but of course, it's very necessary to be very rigorous in others, not to send humo. No, this is science. Next slide, please. We did some research. Real Madrid probably is the best sports team club in the world in terms to win European championships. But sincerely, I believe that Football Club Barcelona probably today is the leader in sports team research. Because in the last 10 years, we have invested a lot of time, more than money, in order to have this research, in order to now to show you. This is that is an example that we are doing now with Omniscope in terms of how change the immunage age in function about your position. Next slide. This is an example. And this is a little bit more that I would like that Holger, can you explain me this last research that we are doing?
No, for us, so what you see is the immune age evolution of a player over the course of a season. And for us, it was incredible to see that immune age now as a metric of immune fitness or fitness in general is changing very rapidly. And it's not changing consistently across the entire team, but there's actually correlation. So players that play more matches, players that move more, age faster. Strikers, so forwards, age faster than midfielders or defenders. And what you see in this slide is the very high resolution picture where we correlate immune age together with GPS data. So football players are probably the best profiled players on the world when it comes to movement. So movement in general, but also impact. And this is all represented by different GPS metrics. You've seen on my slide that we have wearables integrated from the, for example, from the Apple Watch, VO2 Max, heart rate variability. So here this is very different because we have much more different metrics that we can correlate. And for us, it was incredible to see that GPS data, so the external load, is translating to very different internal load of a player. And immune age has been our sensitive metrics to quantify this.
Thank you, Holger. Next slides, please. And what about epigenetics? Of course, we are genes and how we can adapt to how we are sleeping, nutrition, training, travels. Epigenetics, of course, is very important. And probably we need a good balance between genetics and epigenetics. Please, next slide. But today we have the opportunity also to work in terms of the methylation, etc., in order to know this another aspect is how our players have adapted to that training, to the competition. And for this reason also we have another partner that we are working on and researching in terms of to know the epigenetic age clock, the telomere-related age clock. Maybe later another speaker can talk about more on this. But it's interesting to know, for example, that our players, for example, have this specifically in male athletes, in football male, they are a little bit older than the normal population. 2 years more. But in female teams, no. And this is another message. We need more research in terms of female teams. Because in terms of the female, we don't know anything. When we are looking for some biomarkers in terms of transcriptomic, proteomic, metabolomic, they are different biomarkers for female and male. And for this reason, we need to also remember to research in women. Next slide, please. And this is one of the latest studies that we are doing that just only with some epigenetic, we have the opportunity to know the relationship between the load, and some biomarkers that can help us in order to anticipate different injuries. Another slide, please. Fourth, at the end, understanding an athlete's immune, genetic, and genetic profile may help with personalized preventive strategies. This is the key. Sometimes the people say me that, doctor, are you doing a genetic and epigenetical test before hire contracted players? No. But when we contract a new player, I need his permission in order to do this kind of test. But I think that this is good because he knows, and we know, which are the risk factors in different areas. And for this reason, we can create a specific strategic, preventive strategy to help him in terms of health and performance. And the last, next slide, please. This is the coach. Remember, in the wall of the sport, this is the boss. But he believed in this. And we try to explain them to the coaches that a first step towards personalized and precision medicine in the world of the sport is fundamental. It's fundamental. I think that this is my last slide. Thank you very much for your attention.
Dr. Rodas, thank you so much. I think we're uniquely privileged because we can learn the secrets from these elite athletes. And with Football Club Barcelona, we are working really, really closely to bring the best type of science, which already as an accredited FIFA medical center exists, and put this all together. But you see, at the end of the day, I'm certainly not an athlete. So my question to you is, when we look at athletes, do they actually live longer, healthier lives compared to the general population?
Well, this was one of the meta-analyses that in part create evidence, scientific evidence, that this kind of guys, this kind of athletes, of course, have a span life longer than all of us. This is true. For this reason, I think that we need to learn about this kind of special population. ¿Vale? It's true that maybe they will have more problems in their knees or in their hips, okay? But in terms of less disease and long and lifespan longer, this is the answer.
Well, I guess the natural next question is, we see that with a free, non-medical intervention, exercise and sport, we can see scientifically that we lengthen life and reduce the risk of many diseases, including cardiovascular and cancer. But a lot of us in the audience, and even for what you saw on OS Lifetime, using our wearable data, we're not training like athletes every day, right? So in order to get the benefit to live a longer life, do you need to train 8-10 hours a day like an athlete? What do you need to do?
No, but we need to do exercise. And this is another question. It's the last question for you. I would like to know if you can do some exercise every week. How from here, how many of you are doing exercise at least 3 times per week? Thank you very much. You are really, really incredible. Well, but it's not necessary to do 10 or 12 hours per day, no. But that is fantastic. Thank you very much. I think that this is a great population.
Do you want to add anything?
Just a final announcement. So you have seen our both OS longevity profile, but we also want to bring OS longevity to you. So that's why it's our honor to announce today our partnership together with the Sen Clinic in Stockholm and Sweden. So here, I've been visiting Stan Clinic for the last couple of weeks, and it is a perfect match where science meets clinics. So I have been very impressed. The implementation of geoscience and therapeutics, and moving forward, we want to see those lifetime being applied. And Stan Clinic is the perfect partner in crime for us.
So if you're looking for science-based longevity care to increase your health span, together with Omniscope technology, both the Chief Medical Officer and the CEO of Stan Clinic are here in the audience with us today. And I personally think that it is one of the best clinics around in a country which is very much health-focused. So why are we bringing this to you? In the spirit of public health. We don't just want another health tool. We're not just here to take care of 1 or 2 people. We want to make the world a better place before we leave it. And the only way to do that is to invest in yourselves. It's to invest in your own lifetime, to have a technology which allows you to achieve your goals. And today, as a thank you to José, we would like to give one full package of OS Lifetime, which is the same technology which Dr. Rodas presented, to allow you to become part of this community. So grab your phone, grab the QR code, because one individual in this audience today will win your OS Lifetime. And on that note, I wish you a long and healthy life.
Thank you. I was just about to grab my phone because I definitely want to win that, but I guess I have to give it to the audience. But thank you so much for a great presentation. And it's quite synchronistic because I know the Stan Clinic and I was actually thinking, I think they need to meet Omniscope. And here you are having a fusion. So I can definitely verify it's actually one of the role model clinics. So thank you so much. And hopefully I have time for a bit of a Q&A. I'm just going to introduce the next speaker of this session. I'm just going to introduce the next speaker of this session. And we wouldn't actually be able to maybe measure different biomarkers in our body if we didn't have what the next speaker has given us. He is principal investigator at Altos Labs and the scientist and genius who gave us the epigenetic clock, redefining how we measure biological age, and made the seminal discovery of the Horvath clock, an epigenetic clock based on DNA methylation. Please, a round welcome of applause for Steve Horvath.
Thank you so much, Jose, for this invitation. It's been an honor to speak after Mehmood Khan, who has done so much for the rejuvenation field with evolution. I was very impressed by the Omniscope technology. I will talk about epigenetic clocks and also immunity. I think some of my material matches the previous talk. So there are many biomarkers by now. There are broadly 3 categories: molecular biomarkers, such as methylation and so on. Then there are these physiological biomarkers, functional performance, and of course, we are now in the era of wearables, various trackers of step counts and so much more. And I will really focus mostly on these omics measures, in particular methylation and epigenetics. Here I show you a short history of biomarkers of aging, and it goes all the way back to the 1930s, actually with these foundational experiments that caloric restriction in the rat can slow aging as measured by lifespan extension. You calorically restrict a rodent, it will probably live longer. By now we know that caloric restriction is very much a gold standard intervention for slowing methylation clocks. And what this timeline shows in red boxes are epigenetic clocks. Interestingly, when people compiled this history of biomarkers of aging, they listed many, many methylation clocks. And the question is why? And the answer is obvious. Methylation contains a huge signal for aging. That's why. It was easy to do, you know. So in 2011, we published the very first epigenetic clock based on saliva. And we were, of course, hoping that these saliva methylation measures would be useful. And later, if I have the chance, I'll talk about how we measured saliva methylation in a soccer team, Premier League in Germany. Maybe not as good as the football club in Barcelona, but still very good. But anyways, then the multi-tissue clock. And now we are in the era of next generation epigenetic clocks that predict mortality risk. So I was always interested in how to measure biologic age in people who do follow the perfect lifestyle. Here I show you a well-known body weight athlete, bodybuilder, and he, of course, has tremendous muscle strength, but probably he wouldn't be able to fool many molecular biomarkers of aging. Our best mortality risk predictor is called Grim Age. It's named after the Grim Reaper. It does not predict your calendar age. Rather, it predicts in certain ways, your risk of dropping dead in the coming year. That's kind of what it measures. It's a mortality risk estimator. So the field has developed many mortality risk predictors and epigenetic clocks, and their comparisons. Here I show you a comparison of a dozen epigenetic clocks, and Grim Age still stands out as the most significant predictor of mortality risk, which surprises me because this was developed in 2019 and many years have passed. I'm hoping for more powerful epigenetic biomarkers. An early criticism of epigenetic clocks was that we don't understand the mechanism. What exactly do methylation clocks measure? But we now have learned quite a lot. After 12, 13 years of tremendous effort by the whole aging community, we now know many factors that accelerate aging. In short, methylation clocks are integrators. They integrate a lot of various stressors and damage accumulation. But one important aspect of what they measure is changes in cell type composition. For example, the previous speakers talked about the immune system. And I now want to talk about really this relationship between age-related changes in the immune cell composition and epigenetic age, in blood, of course. So Andrew Teschendorf recently published a wonderful paper, very large-scale study, where he accumulated a dozen different large cohorts to ask the question, well, what cell types correlate with various epigenetic biomarkers? And here I show you the results for Grim Age. One insight is people who have a high abundance of naive T cells, CD4 T cells or naive CD8 T cells, so naive, these are in essence young T cells, sure enough their Grim Age is reduced. Conversely, people who have a high abundance of so-called monocytes, of adult monocytes, their Grim Age is accelerated. So these results very much match some of the material from Omniscope, who of course track different immune cells. But yes, so that's all expected to some extent. What this slide shows is that epigenetic age relates both to the adaptive immune system, T-cell biology, but also the innate immune system, monocytes, for example. So when you go to the doctor will sometimes measure a complete blood count and with flow cytometry or related technology. But interestingly, we can really deconvolute blood cell composition just using methylation. We measure your methylation and we can estimate the abundance of many different cell types. Here I show you the readout of 19 different cell types. And now you can ask, well, which of these cell types actually predict your mortality risk, your time to death? And interestingly, one of the top hits are, again, these naive CD4 T cells. If you have a high abundance of naive CD8 T cells, you will live longer. But now there's a question. Okay, we can measure methylation and we can measure blood cell composition. Do the clocks measure anything beyond just blood cell composition? And here I show you briefly the answer. So here we predict time to death even after adjusting for 19 different immune cell types. In other words, we remove the confounding effect of blood cell composition, regress it out. And what you can see is these epigenetic clocks are still highly predictive of all-cause mortality, even after you regress out anything you can. Here I show you that epigenetic clocks predict cancer-specific death, will you die of cancer, or, of course, cardiovascular death, even after regressing out the immune cell composition. So the conclusion of this very large-scale and comprehensive study is that, yes, epigenetic clocks reflect the innate and adaptive changes of the immune system, but this is really not the whole story of epigenetic clocks. Rather, epigenetic clocks capture deeper layers of biologic aging as well. We sometimes call it innate aging. Now I want to come to this topic that was briefly discussed by Dr. Kahn, young plasma. Should you get young blood plasma? There have been studies in rodents, the so-called parabiosis experiments, where people have shown that if you connect an old mouse to a young mouse, then you get benefits in the old mouse. And Vadim Gladyshev and Bohan Zhang and their labs have shown that, sure enough, epigenetic age is reduced if you expose the old mouse to young blood. So conversely, Jesse Poganek and Vadim Gladyshev showed the opposite phenomenon. You take a young mouse, you connect it to an old mouse, suddenly the young mouse ages faster and epigenetic age is increased in the young mouse. And that, of course, leads to an insight or an idea. Well, maybe the old blood plasma contains factors that make you age faster. And therefore, you should remove or filter out these old pro-aging factors. And here I show you the results from a human clinical trial that evaluated plasmapheresis. So plasmapheresis is a procedure where old blood plasma was removed. And in this particular study, Pavel did not replace the removed plasma by anything else, just removed it. I think of it like an oil change. Get rid of the old oil or the old plasma. We were, of course, hoping that this procedure would lead to rejuvenation of epigenetic clocks. And we looked at multiple epigenetic clocks, and there was a frustrating result. It was the opposite of what we expected. Actually, the number of plasmapheresis sessions was associated with increased epigenetic age according to multiple clocks. So it was disappointing. Now, these investigators were very thorough and they looked at many clinical markers, for example, lipid levels or red blood cell distribution and so on. And these results were mixed. So the finding was that, for example, this plasmapheresis, yes, led to lower total cholesterol, but conversely, it made things worse when it comes to red cell distribution width and other biomarkers. So overall, we couldn't see any benefit of using this plasmapheresis for rejuvenating these individuals. Now I want to come to another important topic, metabolic stress, for example, due to obesity. So everyone has heard perhaps of semaglutide or these GLP-1 receptor agonists. These are miracle drugs for helping you to lose weight. And of course, the longevity field has been very interested in the question whether GLP-1 agonists, receptor agonists, would lower epigenetic age. Here I show you the results from a preprint. I was not involved, but I thought it was a very nice study. And pretty large scale, 45 treated versus about 39 controls. And I do want to highlight that this population was not particularly healthy. So all of these people were HIV positive, but more than that, they suffered from obesity, body mass index 32. However, the striking result was that the GLP-1 receptor semaglutide treatment once a week for 32 weeks very much was associated with reductions of grim age and all other epigenetic clocks after you regressed out, for example, markers of inflammation, body mass index, and other confounders. So here I show you the results for different epigenetic clocks from that paper. You see the negative values. All of these indicate rejuvenation based on this compound. But there are many open questions. So would GLP-1 receptor agonists show similar effects in healthy non-obese people? So in other words, people who do not have HIV. And also, of course, the mechanism. So this remains to be seen. I want to now talk about viral stress. And Dr. Khan mentioned earlier hepatitis C. I talk now about HIV. So we now know that HIV positive individuals who take this so-called antiretroviral therapy for repressing the HIV virus, that their epigenetic age gets reduced by quite a lot. So this is a wonderful study from Raghav Sehgal and Albert Higgins from Yale University, where they looked at 52 different interventions, and they ranked these interventions by which ones had the strongest anti-aging effect. And as I mentioned, the top hit was antiretroviral therapy. The second hit is anti-TNF alpha. So some people who have high levels of inflammation, autoimmune diseases, they need to take anti-TNF alpha. Another oldie but goodie that showed up is metformin, well known in the longevity field. And the other thing that surprised me that also showed up is the so-called hyperbariatric oxygen therapy. So these therapies seem to have an effect on epigenetic age. I want to briefly talk about exercise because the previous talk was very much about athletes. So here I collaborated with Zsolt Radak from Budapest University, and he analyzed Olympic medalists and champions, and he found that people who had won an Olympic medal less than 10 years ago, fairly recently, yes, their grim age was slightly reduced, so they were younger than the average population. However, if an athlete had won the medal more than 10 years ago, there was no difference to the average person. And I show these results because they make an important point that we see over and over again, which is rejuvenating interventions sometimes have a transient effect. You know, you're an athlete, your epigenetic age is reduced. If you don't keep it up, it will bounce back. Here I show you a very rigorous study from Germany where the investigators used a wearable to track the step counts in 3,600 German participants. And sure enough, they found that step count was associated with lower grim age. So yes, walking around had an effect. However, as a biostatistician, I looked at the results, and to be honest, I was disappointed in the very weak effect. So for the experts, the p-value was 0.005 in 3,600 people. To me, that's a very weak effect. Some people talked about supplements. I want to draw your attention to a very rigorous study conducted by a Swiss professor called Heike Bischoff-Ferrari, who studied 700 older participants. Everyone was fairly healthy, 71 years or older, average age 75, and they received various supplements. And then they were followed for 3 years. And one supplement, the most noteworthy finding was that omega-3, 1 gram of omega-3 was associated with lower grim age. Now, I liked this study because it was so rigorous. It was a randomized controlled trial. Everyone was completely blinded. So it's really a result one can trust. I, of course, take omega-3 every morning, but not too much. High dose of omega-3 may cause atrial fibrillation, so be cautious. Now I want to come to the topic, how dynamic are changes in methylation? So what's the recommended frequency for measuring methylation? Should you measure it in a continuous way, every week, every month, once a year? And if you open a textbook, it will say methylation is highly stable compared to any other readout. So textbook knowledge would be measured once a year. But now I want to show you the results from a study of a German Premier League soccer team. This was a very interesting study where they collected now saliva samples, not blood, from players before the season, during the season, after the season, before games, after, over 200 saliva samples from 24 members of the team. And here is the striking result. Before the game, let's say, look at the left panel, in athletes, their grim age is roughly 30 years old. These are young men. Right after the game, you see this drop. So that really surprised me. And then after they rested, the grim age bounced back. So that's already a very good hint that vigorous exercise really reduced the grim age estimate by actually a substantial amount. Surprising to me. It was so surprising that I didn't believe it. I thought this must be an artifact. Now, the first thing I could think of an artifact would be that there's some blood collection problem. But in this study, they also looked at the saliva samples of the supporting staff, so people who didn't run around for 90 minutes. And there was no difference, interestingly. And here I show you the most controversial finding of the whole study. So this is 24 hours before the game, they collect saliva. Right after the game, they walk off the field, they collect saliva in these Bundesliga players, and then 24 hours later, they again collect saliva. And what you see is a substantial decrease in the grim age, in the biologic age estimate. So this really puzzles me, this finding, and I'm trying to understand how that is possible. So I think it reflects that the DNA in the saliva, of course, reflects the blood cells. And this very rigorous exercise must have an effect on the DNA that makes it into the saliva for whatever reason, you know. So we see this transient rejuvenating effect. What it shows us is when we use these epigenetic clocks in, let's say, human clinical trials, we need to be careful that these people didn't just return from the gym, you know. We need to standardize the collection of the DNA, you know, in these athletes. I want to now talk about some more futuristic ways of rejuvenating or slowing aging. There was a wonderful study from Harvard by Dr. Vratin, and he has a way to cool the body temperature of mice. And how does he do it? There's certain neurons in the brain, and if you stimulate them, that leads to a drastic reduction of body temperature in the mouse. And the graph shows you how methylation age in these mice relates to the age of the mice, how it changes. The blue curve represents the cooled mice. And the stunning finding is that the mice whose body temperature was cooler, their methylation age was much lower, interesting. So there's clearly a connection to body temperature and epigenetic aging. I mentioned it, I show you here some AI-generated picture representing science fiction. In space travel, they always put you in hibernation. Maybe that type of intervention could slow epigenetic aging. It's all science fiction. I looked at the effect of sex. It turns out that women age a little bit more slowly than men. And we recently looked at so-called Klinefelter syndrome. And we found, again, a similar finding that men who happen to have one extra X chromosome, their Grimm age was lower than men that have only one X chromosome. So it was kind of an interesting result when it comes to sex differences. Yeah, here I show you the Grimm age results for the so-called XY males versus XY males, but we also looked at many other dosages for sex chromosomes, so I invite you to read the paper. Next slide. Next slide. Next slide. I'm trying to make up for time. One take-home message is for you, there are many ways of measuring biologic age, but they really capture different aspects of aging. I like, of course, methylation because there's this nice idea that the molecule that carries our genetic information also keeps track of time of the organism. These methylation clocks are ready for human clinical trials. People are already using them in some clinical trials. They can be used for studying rejuvenation in preclinical models. I've shown you the study in mice. We have methylation clocks for all mammalian species. I can measure methylation age in dogs, cats, pigs, anything. Here's my acknowledgement, and I'll stop here. Thank you so much.
Thank you so much, Steve, really giving us a deep insight into all the different modalities when it comes to age clocks. I'd just like to call down the other speakers so that we can just have a quick, you always run over time when you're having fun and getting a lot of information. So could I please have Gil, Vijay, are you all here still? Holger? Gil, you're here, that's good. So, here, gentlemen. First of all, any questions? I know it's been a long round and a lot of information, but this is actually correlating both to you as professionals, but also, as human beings, because we all, I presume, check our biomarkers with different epigenetic clocks. Yes, please, if we can just, could someone just help me run with the microphone, please? Ah, Jose, of course. Your Grimm age is going down.
Thank you. So, all fantastic talks. This question is for Stephen. Really great results and really inspiring talk. I was wondering, because we know now that our organs can age at different rates. So then to what extent measuring the epigenetic clocks and all of these tests can recapitulate, if you do it from peripheral samples or from the blood, saliva or whatever, to what extent they can recapitulate what is happening in the organs. Do you think we need also organ-dependent tests, or what is your opinion?
Yeah, so the dream is to take, let's say, a blood sample, measure methylation, and then arrive at results for the different organs, anything kidney, lung, heart, even brain. And the really stunning result is, and I can say this with great confidence, the blood methylation very much captures that information. So there have been several publications out, some as late as last week, but several publications describe now ways of measuring what they call organ ages based on methylation, And I briefly talked about Grimm age. I didn't mention it, but it's based actually on estimators of, for example, C-reactive protein, hemoglobin A1C. Many traditional biomarkers of organ function can actually be estimated with methylation. So many of the experts know proteomics has been used to estimate organ ages. All I can say, you can do very similar things based on methylation. And my thoughts are both readouts will really be needed to capture aging. Not just proteomics, not just methylation, really both.
Thank you so much. I just want to mention we have around 400 people following us on YouTube, so we also have questions from YouTube, and they can vary from really scientific ones to maybe more joy span ones. And one, before I take your question, was to Omniscope. Are you making sure that FC is beating, Barca is beating Madrid, or the other way around?
I think that's a tremendously cheeky question. I would say that in the end, I think, we're bringing science into sports, and we work with a number of teams. And the reason that we do that is because science and sport is about all of us. It is literally, these are two disciplines where the whole world comes together and gets incredibly, incredibly passionate. The reason that we started our journey in Barcelona with Football Club Barcelona is because we could not answer what health meant at a cellular level. And it happened that Holger and I were in our lab very late at night, having a bit of a debate, and we looked out of our window at the Camp Nou, and we saw the players enter, and we had our eureka moment. We said, that's what it means to be healthy. But I think actually you are all athletes, because you are all playing the beautiful game of life, whether you are with FC Barcelona or Madrid or wherever you are. And humanity has been practicing sports for as long as we've existed. So there is complete impartiality from that point of view.
Very well put and very good answer. And also really interesting to really highlight this fact with science and sports and how it can really, catapult both innovation and also incentivize, hopefully, humanity to step up and be maybe not athletes, but athletes in their own arena. Yes, please.
So there are many things that we would like to try that are being sold. There is a lot of blah blah, but of course we need to test. And usually things are done in the clinical trials, and it's expensive, and it's not available for all the biohackers, all the community to try. Yet it would be great. So the cheapest things are things like grip strength or spirometer, or some exercise at home to step from a chair and count your heart rate and things like that. Are these fine in order for the community to start establish, I would say, weak signals? Or should we have a complementary? What is good for what? That's my question. And maybe I will complement it. Isn't it all this, in fact, short-term effects on health? Shouldn't we have animal tests, for example, to have a long-term view? Or is it sufficient?
Looking at a democratizing health level, what is sufficient and what is not, Steve, as the first question?
Yeah, I briefly mentioned it in my talk. These are so-called functional tests, such as walking speed, grip strength, and they are of utmost importance. They should always be measured. I would say it goes without saying that you would mention it. For example, how often you can get up and down from a chair in, let's say, 1 minute. Everyone can do that at home. That's a nice competition. So these are very important readouts. They should be measured. It's rather the other way around. Should you also add genomics, which can be expensive and
There's so many questions to answer, and time is running out, and we're talking about moving.
And this is really looking at what is possible today, what is impossible today,
and what is plausible in the future when it comes to longevity. And we're going to start with Michael Ringel, who is Chief Operating Officer at Life Biosciences, a biotech company focused on partial epigenetic reprogramming to reverse age-related diseases. So please give it up for Michael Ringel.
I'll use the handheld. Perfect. Awesome. Hi, everyone. Yeah, so Michael Ringel, Chief Operating Officer at Life Bio. That's David Sinclair's company out of Harvard. I'm a biologist by training. I actually spent 25 years at a company called Boston Consulting Group, which is one of the large consulting companies. And that's really my origin story, because I did a project for a client who was interested in this space, and I did a scan of all the technologies out there, and I just became convinced that partial epigenetic reprogramming, which I'll tell you about, is the philosopher's stone. It has tremendous potential to address age-related disease. So I bet with my career, quit my job, jumped to the company, and I'm going to talk to you about why you should also bet, maybe not with your career, although maybe, but certainly with your time, with your capital, with your intellect, capabilities, with your heart, and maybe all of the above. So I'm going to talk about this in 2 chapters. If you could click forward, I'll just say click. So click, yeah, and one more, click. So 2 chapters. I'll spend a bit of time on, we'll see. Yeah, perfect. It's working. I'll spend a bit of time on why we need a breakthrough, and then I'll probably just spend most of the time on why I think we found it. So a great starting point for why we need a breakthrough is this slide. This is a survival curve in humans. It's also called a Kaplan-Meier curve. You can see it starts at 100%, it goes down to 0, and it's shown here for 4 different cohorts. And actually there's been tremendous improvement in the last 150 years, largely due to sanitation, vaccination, and reduced infant mortality. But there are 2 big gains that are missing. One of which is health span, which is the quality of those years. We can expect to spend maybe a decade in ill health, which obviously we would like to address. And the second is that despite the improvements in average lifespan, we have not improved maximum lifespan. That's true no matter how much you spend. So here's a chart of health-adjusted life expectancy against healthcare spending per capita. Each dot is a country. And you can see there's something like an asymptote here. And whenever you see something that looks like an asymptote, you should ask the question, are we actually running up against a limit? And the answer is yes, that is the limit. Jay Olshansky and colleagues did a great piece of work published in Science in 1990, showing that if we wanted to get to average, and this is the US data, but average lifespan of 85, 90, 105, we would have to reduce mortality by 55 up to 90%. And that's in the context that if you cured all major degenerative disease, you would only reduce mortality by 75%. So they looked at that data and said, the chance of us radically improving is limited at this point, and so they made the conclusion you see on the right-hand side, which is the period of rapid increase in life expectancy has come to an end. They took a ton of criticism for this, but they've been absolutely proven right. It's leveled off throughout the developed world, and in fact, in some places, it's regressed, largely due to obesity. But this is not their actual quote. What they actually said was unless. They said unless we can address aging at the molecular basis. And they go on to say that they're actually very optimistic about that. So why is that so important? So it's important because of something called the geroscience hypothesis. So the number 1 risk factor for more than 90% of what kills us in the developed world is age. You can see here shown are plotted cardiovascular disease, dementia, cancer, diabetes, really the scourges of humanity. They all go up with age, and they go up exponentially with age. We haven't thought of your age as an intervention point for these diseases because historically we thought you live 1 year, you age 1 year. But if, in fact, it becomes possible to separate biological age from chronological age and actually move where you are on this x-axis, a single intervention can address all major degenerative disease simultaneously, exactly Jay Olshansky's challenge to us, and thereby extend both lifespan and health span simultaneously. And there's a lot of evidence this is possible. We see it in genetic variants. This is an example from worms. This is another survival curve or Kaplan-Meier curve. Worms with a single mutation in a gene called DAF2 in this case live twice as long, and they're also healthier all along the way. They move better, their diseases of degeneration are moved out. We see it in pharmacological interventions. This is an example from mice of the efficacy of rapamycin. This is done by the National Institute on Aging. They have something called the Interventions Testing Program, ITP, which tested various interventions. And you can see here another Kaplan-Meier curve. The mice live longer, and again, they're healthier along the way. Things like cancer is delayed. They're more vibrant as they're living. And then lastly, we see it in epidemiological data. This is from humans, maybe most relevant to us, obviously. And this is different populations of Japanese, from sumo wrestlers to mainland to Okinawans, who are probably the most famous blue zone, along a gradient of caloric restriction, which is the really best validated intervention in other mammals. You can see they live longer, and indeed, again, are healthier all along the way. Great news. The problem is we really haven't advanced on the state of the art in a long time. So the interventions that have the best effect, largest effect size on lifestyle is caloric restriction. We've known about this to some degree since antiquity, and certainly scientifically studied it in the teens, 20s, and 30s. The most effective therapeutic in mammals and in mice remains rapamycin. It was the first one discovered, and it's still the gold standard. Another way to see this is to just compare modern advice to advice from literally 400 years ago. So there's this great study on the left done by Wynn and colleagues at the Veterans Affairs in the US. And it's actually a really good study, so I don't mean to pick on it. They identify the 8 habits that are associated with living 24 years longer on average. And you probably want to know what the 8 habits are. So there's the holy trinity of diet, exercise, and sleep. There's the 3 thou shalt nots. Don't smoke, don't drink, don't do drugs, don't have any fun, right? And then the 2 around mental health, managing your social relationships and managing your stress. But you can go back to Sir Francis Bacon, as I did, and literally find every single one of these in his writings, including some things that you might think of as somewhat modern, like opioids. He says, no, poppy juice will make the spirit curdle, or smoking, right? Tabatha, don't like poppy, buzzle, and trouble. But they're literally all in there, including some things that we talk about now as very ultra modern, like this idea of parabiosis and blood transfusion. He had an opinion on 400 years ago, and I thought it wasn't going to work actually. Which Steve Horvath's data actually supports. So to understand why we've stagnated with these interventions, you have to actually understand why they work. And there are various schools of thought on this, but I'm going to tell you the correct one. I don't have time to explain why it's the correct one, but it is. And the logic is based on evolutionary biology, and it goes as following. So energy and resources are not infinite. So organisms have choices in how they deploy that across a lot of different things. But you can kind of grossly simplify that to growth and reproduction versus maintenance and repair. And when you're in a bad condition, like caloric restriction, like why would starving an organism actually make it live longer, right? It seems paradoxical. And it's not just starving, like many other stresses. Steve showed you cold stress, heat stress, hypoxia. Lots of stresses cause organisms paradoxically to live longer. Well, when they're in a bad situation, so bad that offspring are going to be challenged to survive or thrive, you shouldn't invest in growth and reproduction. So even though your total resources have gone down a little bit during this famine, you over-reduce the amount that you put in growth and reproduction, which means you have a little bit left to shift to maintenance and repair, to wait out the bad conditions and wait for another breeding opportunity. And if you're a worm waiting for a rain, basically, a few extra days actually matters a lot, or a fruit fly waiting for a fruit to fall, extra month matters a lot, or a mouse, one extra breeding season matters a lot. And there's a lot of evidence that this is actually what's going on, including many of the key genes involved are actually what are called diapause genes. DAF2, which I showed you, were kind of kicked off the field, is a diapause gene. It's involved in creating this quiescent state where organisms try to wait out bad conditions. So you can plot all these known interventions against a master switch that exists for this plasticity to shift between these states. So a switch should be able to sense whether the conditions for growth are ripe or not. So are the building blocks present? Do I have oxygen? Do I have a template suitable for growth? Crucially, do I have energy? The switch exists. It's called mTOR. It integrates all these signals. And that switch should then turn on or off growth and reproduction. I lost my screen down here, but I'll look back up there. Or the maintenance and repair, which are shown in red. So you've got turning on protein, lipid, nucleotide synthesis, turning on mitochondria, which put energy in a usable state, turning off these repair and recycling pathways. And again, you see exactly that. And we can plot the known interventions on this. So remember rapamycin, the intervention that had the largest effect size ever shown in mammals? Well, it sits right there. The next two in the ITP that have the largest effect size on maximum lifespan sit right there. Everything else that's ever passed the ITP sits here, including new information from just a week ago. And, there's a few others that people talk about. And, if I've missed out your favorite one, I'm sure there's a spot for it on here somewhere. But when you go back, when you have this understanding of what's going on, you go back to this slide, you understand that this mechanism matters a lot more in a short-lived organism than a long-lived organism. So that fruit fly waiting for the fruit to fall, a month matters. But if you're already living 80 years, you're actually averaging out a lot of this environmental variability. And so the prediction is that caloric restriction and these other insults and these other mechanisms matter more in short-lived organisms. That's exactly what we see empirically. It's a little bit of a fanciful chart here. I've put it together from a bunch of different sources. They've been done in different methods and so on. But you can see this inverse log linear relationship between how long the organism lives naturally and how much benefit it gets from caloric restriction. And you get down to humans and you suspect that in terms of maximum lifespan effect, we're probably only talking about a couple of years from a lifetime of caloric restriction. It's also, by the way, supported by the epidemiological data. Okinawans are pretty close to the limit of what you could minimally eat, and yet, they're not living to 150. The implication here is that the best interventions that we have right now, they're worth doing. Don't misunderstand me. You should do every one of those 8 healthy habits. They will make you healthier in the years that you have. They will cut out 24 years of early mortality on average. They're really worth doing, but they're not going to make you live past 120. But there is a different kind of biology that has the potential to reset that, because it taps into a totally different mechanism from what I just showed you. It's the biology that makes babies young. So there's a picture of my wife when she was 37 and my youngest son. At the age of 37, her egg cells were pretty old. And even at 18, right, she's an adult, not an infant. When an adult has a baby, that baby just comes from their own cells, their old cells. So there has to be a mechanism in nature that takes an old cell and makes it young again, which is a profound point, right? We all know that babies happen, but this idea, somehow it just comes from an old cell. Well, there must be a mechanism built in that makes them young again. We actually now know what it is. Between day 7 and 9 of embryogenesis, the epigenetic marks that Steve Horvath talked about, these methyl marks, acetyl marks on the DNA, on the histones the DNA is wrapped around, are just wiped clean. And the cell from that point forward is told it's young again and behaves like it's young again. So if the parent had Alzheimer's disease, or the parents had cardiovascular disease, or the parent had diabetes, the baby does not. It might have some sequelae from that. If the parent had a genetic disorder, it can be passed on. But in general, the epigenetic disorder is not passed on. The age itself is not passed on. That's been tapped into artificially. Shin Yamanaka won the Nobel Prize in 2012 by showing that you could treat cells artificially with just 4 small proteins, abbreviated OSKM. João Pedro may talk about it a little bit. Those 4 small proteins take an old cell. I could take an old skin cell from one of you and turn it into a pluripotent stem cell. And in this discovery, people focused on the fact that these cells were pluripotent. They could become anything. They no longer had an identity. They were no longer a nerve cell, a muscle cell. They could become anything. But hidden in that discovery was the fact that the cells were completely rejuvenated. So if you grow a clone from a cell that's gone through this process, the clone is not born as an 80-year-old individual. The clone is born as a baby. Now, it's not ethical to do that in humans, but in mice, for example, this work has been done. So the thing that people find conceptually hard to believe, that we can actually de-age a cell, we actually already do. It's known technology. It's routinely done. It's in the context of this reprogramming. Now, the problem is you don't want to do that in your own body because you don't want to take your cells all the way back to pluripotency. You would cease to function. If you do that in a mouse, you'll kill it in about 3 days because your nerve cells have to stay nerve cells, your muscle cells have to stay muscle cells, otherwise you stop functioning. There's also a risk of cancer. But the biggest insight that came out of the Sinclair lab that was on the cover of Nature in 2020 is that if you treat with just 3 of the 4 factors, abbreviated OSK, like it's so simple, right? Just drop one of them, see what happens, right? You take cells partway back. They get younger functionally, they get younger transcriptionally, they get younger epigenetically, but they don't go all the way back to pluripotency. So it's safe. And this is the study they did that was on the cover of Nature, which was in eyes in mice. It's actually a picture of an eye with like a clock going in reverse, where they restored vision to blind mice. So that's the technology that we want to take forward. You could use that in any age-related disease, theoretically. And here's a list of just examples from the literature showing different diseases associated with epigenetic changes. It's literally every organ system. But you have to start somewhere, and so our company, Life Bio, is taking David Sinclair's insight and starting with the eye, 2 major forms of blindness, glaucoma and nyon. So the number 1 cause of blindness in people over the age of 60 is glaucoma. The number 1 acute cause of blindness is nyon. There's really tremendous unmet need here. If you catch glaucoma early, it's actually pretty well treated, but unfortunately many people catch it too late. There's actually 3 million people already blind, so a huge amount of unmet need that cannot be treated. And nyon actually has no treatment. We're taking our first molecule forward. There's a tremendous preclinical data package now showing that this works not only in mice, which was the original work done in the Sinclair lab, but now we've repeated that in non-human primates, monkeys, showing the full logic chain of what you would want to see. The drug is getting to the right cells. It is actually shifting their epigenetic state right back to youthful. It restores visual function, so the electrical impulse that comes out of the nerves in the eye goes from when we blind the animals, becomes flatline. After treatment, can go all the way back to complete youthful signal. The 4th panel there is actually a section of the optic nerve showing the axons literally regrowing back down the nerve. First time nerve regeneration has been shown. We show visual improvement in the mice. We can't actually test it in the primates, but everything else suggests that we would see that in the primates. And importantly, in the studies that have been done, it's been seen to be safe. And they saw no nanog, which is a marker of going back to pluripotency in the mice work. They saw no increase in tumors. And I'm happy to report we've now finished our GLP tox, which is the preclinical work required by the FDA, and had no serious adverse events. So we're going into humans very shortly, Q1. We'll be testing in actual patients. So even though it's a phase 1 study designed for safety, we'll get some efficacy data, ideally by the end of next year. So it's going to be a huge year for Life Bio, obviously. It's actually a huge year for the whole field, because it'll be the first time ever a rejuvenation agent has gone through clinical trials and hopefully shows efficacy. So, watch this space by the end of next year. We wanted to demonstrate that this is not just an eye effect, so we've been working on other organs. We haven't disclosed all of that, but we've disclosed one, which is liver, which is about as different from the eye as you can imagine. So the eye is compartmentalized, there are neurons, they're not dividing. The liver has a ton of crosstalk with other organs. It's one of the most regenerative organs. And yet the same drug, the same underlying mechanism of action addresses fatty liver disease in these mice models. So you might ask, why would a glaucoma drug work on fatty liver disease, right? That would be a paradigm that would make no sense to a physician coming at this from kind of a historic way of thinking about medicine. But when you understand, well, if I rejuvenate the cells, they actually can heal themselves, it becomes a very powerful tool to address all age-related disease, and now we've demonstrated that it works across organs and diseases in mice so far. So that's just a snapshot on the company. It has this technology that can rejuvenate cells. You could address any age-related disease with that, potentially, aging itself perhaps. We're about to enter clinical trials, expected Q1 of next year for these optic neuropathies. Many other diseases we'll go after. And we actually are in our Series D financing. We're a bit over halfway done with that. Anyone who's interested in more information around that, or just life bio more generally, obviously here's a QR code where you can link with me, and very happy to take questions. But I think we're going to do that at the end of the last session, right? Some now?
No, we'll do
at
it
the end.
At the end.
All right.
Thank you so much.
Thank you very
Please
much.
give it up for Michael. Thank you.
Thank you so much, Michael.